This is episode 77 of the Immunology Podcast, Computational and Translational Immunology with Dr. Caleb Lareau. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Caleb Lareau from the Memorial Sloan Kettering Cancer Center on the podcast to talk about his research on computational immunology. We also got our usual roundup of recent highlights of immunology news coming up. But first,
the AAI 2024 annual meeting will be held May 3rd to 7th in Chicago, Illinois. AAI President Akiko Iwasaki will kick off the meeting on May 3rd with her address, Learning Immunology from Viral Infections. Dr. Iwasaki was also an organizer of a special symposium focused on the immune system and in tissue homeostasis and disease. Symposium speakers include Jasmine Belcade, Susan Keck, Diane Mathis, and Mark Jenkins. So very impressive lineup. Don't miss this exciting lineup of top researchers in the field. Visit immunology2024.aai.org for more information.
Well, it's almost here.
Yes, almost here.
And I booked all the things like I promised, so we're good.
Very good. Very good. I'm looking forward to our restaurant selection. And you know what? What else are you looking forward, Jason?
So one thing I am very excited for is, we'll get to this
the live show.
Yeah, the live show. Like, look, we got a live show going. It's going to be May 4th, 12:30 to 2 p.m. Topic is artificial intelligence and immunology with guests Joe Vinny and Ryan Peckner from Seismic Therapeutic. But it's a live show on Star Wars Day. And I just promise everyone, no details. You got to come to the live show to see it. I am bringing my A game, and the Force will be with me and all of you who come.
So, as we said, May the 4th at 12:30 p.m. to 2 p.m. That is our slot. We hope that you listeners will join us in this conversation, and we'll see how it goes, how it comes up. It's first time. I'm so excited.
There's a lot of firsts. We also have the other podcast, Lab Coats and Life, which is a whole expansion we've been doing. So That's been exciting. So people should check that out.
So we have already, how many folks? So we had like 4 episodes out already?
Yep, 4. It's things like GP a postdoc, how to go from a mentee to being a mentor, inclusion and belonging in science, how to go from bench to bedside and get studies from the lab into the clinic.
Mm-hmm. Your specialty.
Yep, exactly. Got to get it in people somehow.
Very nice. Very nice. So yeah, so don't worry, for our listeners, you can find them also on wherever you find this show. You can find Lab Coats and Life podcast.
Well, Brenda, one of the things you don't want to get into people is influenza. But when you do, it becomes a problem. And with that is my segue into our roundup.
Oh gosh, worst segue ever. Give me a break, Jason. You got to bring your A game to the conference. All right, bring it. Impress
so talking
me.
Influenza in Nature, the article is Necroptosis Blockade Prevents Lung Injury in Severe Influenza. And first author is Avishek Gautam, and last author, there's 4 corresponding authors: Gregory D. Cooney, Paul G. Thomas, Alexei Degterev, and Siddharth Balachandran. So it came out, was received the 10th of February, 2023, was accepted March 1st, 2024.
Not too bad, just a year.
Published the 10th of April, 2024. Thank you, reviewer number 2 or 3, depending on who. But we know how that goes. All right. So when you get influenza A, you can get something called ARDS, acute respiratory stress syndrome, which is this cytokine storm mediated process with tissue death, and your lungs fill up with pus and fluid, and then you die, and nothing we've ever used gets it better. You put someone on a ventilator and you try to write it out, but anti-TNFs, steroids, we've never gotten anything to work for ARDS. There's a whole study called ARDS-NETs. There's all these trials. We tilt at windmills and we all suck. Can't fix it. You just got to write it out, put the lungs on a vent, hope they recover and they can come off of it. But the ventilators cause lung damage. So it's kind of this doom spiral and it sucks. And that's why influenza kills people, basically. So that being said, one of the things that happens with influenza is something called necroptosis, which, remember, got apoptosis, which is happy programmed cell death. Then you got necrosis, which is just it dying. Then you have programmed necrosis, necroptosis. We also have our friend from Gastronomy D, pyroptosis. It's relevant in this paper because they check. So one of the main activators of this process is zinc forms. There's a zinc Z-form nucleic acid binding protein, ZBP1. That is a extracellular sensor for influenza A virus infection. And that activates something called RIPK3. RIPK3 then activates apoptosis, and it also activates necroptosis through MLK-like protein, MLKL. And so you can use things like an apoptosis inhibitor, an MLKL pharmacological inhibitor, and shunt the pathway one way or another, or do things to understand apoptosis or necroptosis. Now, if you happen to kill, if you inhibit these too much, you can actually shunt things and have runaway apoptosis, which could be germline lethal or inducible. So they make inducible versions, right? So you can do an MLKL inhibitor, but like doing a RIP3K knockout, you tend to either have to you can do that, but you have to be careful with some of these pathways, because if you just shunt it from one to the other, you can make even embryonic lethal strains by having, say, runaway apoptosis in the absence of the counterpath. So just to know that, like, there's these shunts from side to side, and so the signaling gets complicated, and so you have to use tools like pharmacological inhibitors that also shut off apoptosis or what have you. So RIP3K has a kinase. The kinase-dependent pathway phosphorylates MLKL, that's necroptosis. Non-kinase dependent activity of it, so it gets activated, but when activated, it activates caspase 8, and that goes under apoptosis. So other people, including GSK, and this paper talks about that, have done inhibitors of RIP3K that specifically go after the kinase, right? So you're only going to go over at the necroptosis and not the apoptosis. And that's important because if you shut off both with just a pan inhibitor or knock it out, mice die from flu just as bad as wild type or worse. Because what you want to do is turn off the necroptosis so that your lungs just don't give up the ghost and die. But you want apoptosis to occur so infected lung cells that are really sick undergo apoptosis, but not everything in an inflamed environment is going under necroptosis, right? Because ARDS basically is like, instead of just killing the lung cells that are infected, let's kill all the lung cells. That's kind of the problem. And all the lung cells in necroptosis versus the ones that are infected. So they just capitulate some of the importance of the knockout and the virus, these viruses. But this is a kind of redone work that's the first step up of, they're saying, like, look, in our hands, an MLKL knockout is protected from the virus. Okay, fine. But they are just recapitulating. But they talked about these GSK drugs that kind of worked, and they took a different drug, which was actually a BRC-ABL kinase inhibitor that had known to bind to RIP3K in previous literature, and then did more changes to it through, chemical engineering to come up with something that was super good at binding RIPK3. They do a crystal structure of it. They show how it's better. They show why. And then for the rest of the paper, very succinctly, they show that it blocks necroptosis only up to pretty high doses. If you go really high, eventually it affects apoptosis too, but you're way above the level of therapeutic effect you need to block necroptosis. And guess what? If you block necroptosis, lethal doses, 80% of mice survive. And you can administer it with the virus. You can administer it after the virus has been administered, like in real life, 48, 72 hours later. So you can wait. I'm infected. I'm sick. Now I take this, and this still recovers. And so it's basically showing as a way for lethal, and then they also show sublethal influenza in mice, and then in human cells and in human lung tissue, but not in people, obviously, that this works to prevent viral infection and mice up to including death, weight loss, everything you'd expect. Lung pathology, fluid in the lungs, cytokines. It works. It prevents necroptosis from happening. So it looks like a pretty good drug to go after now. And they do some preclinical talks and say, look, like we don't see these things at this level of dose. We don't see lung issues, kidney issues, liver issues. So which is kind of what you want to start with to say, hey, I want to put this in people. And so now you'd have to figure out how to formulate it for people. You'd have to do maybe a monkey study, and then you could get this into people in a phase 1. For people who are sick in an ICU or something, you can get this into pretty quick because nothing works right now. So throwing this on board with a decent safety profile is a pretty good trial. So there you go. They're starting to come up with anti-ARDS things by going after necroptosis. And the GSK drugs failed because in cell systems, they weren't as good as they were in some in vitro assays at actually inhibiting the necroptosis. So it's the same principle that pharma's gone after, but this drug's just better.
So the obvious question is it only applies to influenza models, or can you, or is this ARDS syndrome also the same what you see in like COVID or other respiratory diseases?
Well, fun question. COVID doesn't have a huge ARDS component. When we first saw COVID, we were treating it like ARDS, putting everyone on vents and blowing out their lungs. COVID has a thrombosis component to it where there's a lot of blood clotting that goes on in the lungs. And so you have what's called a VQ mismatch or a perfusion oxygenation mismatch. And for that, you just do BiPAP, so CPAP, BiPAP machine, push air everywhere you can, but that doesn't cause lung injury like a vent does. And so you actually don't want a ventilator in COVID, generally speaking. That's why flipping people prone in COVID was so effective as well. And steroids work in COVID to tamp down the thrombosis process and that inflammatory cascade, and it doesn't in ARDS. So other diseases do have ARDS, but the murdering disease of humanity for ARDS is influenza, or then influenza with a secondary pneumonia. But influenza causes ARDS and makes people dead. Interesting, COVID doesn't kill you through ARDS, generally speaking. Gets you other ways.
And so the thrombosis in COVID is because of the expression of the receptor
on endothelial cells, and then that whole thing. Remember, there's the blood, and even the vaccine can cause blood clots and other stuff. So it's part of its pathophysiology. But treating ARDS for COVID was actually a mistake that myself included did, because that's what we were,
we're told
in the early days in March, that's what we were doing. By May, we knew that didn't work.
Very interesting. I mean, influenza can be very deadly too. So it's good that we have this kind of therapies to reduce the damage, because in the end, right, a lot of it is you're killing yourself trying to fight out the virus.
Yep, you're going down with the ship.
All right. Okay, so for my first story of the day, I will bring you to the magical world of the germinal centers. And we and this study looking into the cues that B cells get in and around, their GC transformations to know when they have to leave the germinal centers that become memory B cells. So the paper is called Interleukin-4 downregulates transcription factor BCL6 to promote memory B cell selection in germinal centers. Was published in Immunity on April 9th. And first authors, Laila Shihata from the lab of Marion Pepper at the University of Washington School of Medicine. And basically, this what this work is trying to explore is how does IL-4, what is the role of IL-4 in helping B cells navigate their time through their germinal centers. So, just the basics. We know that B cells have to undergo rounds of selection and affinity maturation. That happens in the germinal centers, which are these highly structured, highly organized structures that are found in B cell follicles, in the lymph nodes. And during their time in the germinal centers, B cells proliferate. They undergo somatic hypermutation, in which they diversify their B cell receptors. So they start with some kind of they enter the germinal center with their terminal coded B cell genes, and then this get mutated. And the idea is that those B cells with an increased affinity are going to survive more, and then you end up having B cells that are recognize the antigen much more strongly than the original ones. But then this means that at some point B cells have to leave the germinal center because they need to go and become memory cells. Or so in this case they're speaking in particular about memory cells. This doesn't apply so much to plasma cells that are also a product of the germinal centers. But the idea is that there are signals that are indicating these B cells, what they have to do. One of the things that are amongst the known cytokines or the well-known players in this are IL-4, which is a cytokine that is produced by NKT cells and follicular helper cells, which one of the main contributions to these germinal centers is that they promote expression of BCL6 in B cells, which is originally not expressed on kind of naive B cells. And this BCL6 expression is characteristics of B cells undergoing germinal center maturation. And BCL6 is important. It prevents, so it's expressed both in B cells and in T follicular cell helper cells. It's thought to prevent the terminal differentiation of B cells, and it's an important survival signal for B cells that are undergoing this mutation. It works as a anti-apoptotic factor that suppresses DNA damage sensing, and this is important because these B cells are suffering constant rounds of DNA damage. So this is part of the things that keep B cells alive through the process. However, BCL6 downregulation is required for B cells to kind of exit the germinal center and become memory B cells. So memory B cells have a reduction in BCL6. It's not clear what is the relationship. So there seems to previous work suggested that there's a relationship between IL-4 and BCL6 expression of the cells that are exiting the germinal center, but it's not clear how this works. So this is what the authors kind of went down to do, and they looked into a model of Plasmodium infection in mice, in which they could track antigen-specific CD4 T cells and B cells as well. And what they see is they look into IL-4 and they say, well, in an ongoing immune response, you see that most of the IL-4 is being produced by follicular helper cells in the germinal center. So there's a lot of IL-4 being produced in there. And that B cells are expressing a receptor for IL-4 very early on during the immune response. And this at least at the beginning, the IL-4 production by this follicular helper cells encourages the formation of germinal centers and the expansion of antigen-specific B cells. But then they start kind of looking into they try to find out what does IL-4 do at different stages. You have the germinal center start, but at some point germinal centers also become smaller once the infection is done. So there's a certain kinetic to the development. And they see that although early IL-4 encouraged germinal center formation, if they have in a model in which they add IL-4 in the form of a IL-4 complex, and they kind of add excess IL-4 at different times after infection, they see that if they add IL-4 at day 12 to 14, so reaching the 2 weeks after the start, this actually reduces the size of germinal centers, and it alters the amount of B cells that are exiting these germinal centers that are specific for the antigen, where they're looking for a particular antigen of Plasmodium. So what they find is that actually IL-4 treatment in this window, or excess IL-4 in this window, decreases expression of BCL6 in the germinal center of the B cells, and this actually has a negative effect on the survival of of memory B cells with high affinity. They find that indeed, although IL-4 can increase the expression of BCL6, if this goes over a certain threshold, if you have an excess of IL-4, then BCL6 actually has a kind of negative feedback loop. And then they drop this generates B cells that they lose BCL6 expression too early on, and then they don't have the survival signals anymore, and they don't have enough of other survival signals to allow them to survive and become proper memory B cells. Sorry. One of these other survival signals is expression of BCL2, which is a characteristic of memory B cells. But then they don't have this because they don't develop enough. They are not ready to kind of leave the terminal center, and they don't have these other signals, and then you end up having more cell death uh, in this very critical stage. So they find that basically IL-4 signal. So this is through STAT6. So it's like proper canonical signaling of IL-4 leads to this BCL6 negative autoregulation, and this enhances the exit of the from the germinal center of cells that are still not ready to become full-blown memory B cells, and this basically uh, generates the cell death that limits the formation of germinal center derived memory cells. So it's very interesting because it's always this very, you can see that cells are kind of navigating a very delicate balance, and it's very interesting how you see that if you have too much of something, you actually can be very detrimental.
Yeah. So why would you want that negative feedback loop then evolutionarily?
Well, I think that the idea is that normally you have a certain amount of IL-4 that is determined by the follicular helper cells, and so this signaling is it's you don't expect a large increase at the end of so in principle you have this specific IL-4 formation which is necessary for the germinal center to happen. And normally, the normal stage is that B cells start downregulating. If you don't have, so they start downregulating BCL6 after they have enough of other survival signals. So it is not normal. So that yes, you don't expect it to be so much IL-4. This is if you force the system to have excess IL-4, you have this effect. But in principle, the normal thing is that B cells receive other signals that allow them to leave the germinal center, and this happens when and normally a reduction in IL-4 will reduce the amount of BCL6, and then that's part of the phenotype of memory cells that have exited the germinal center.
Speaking of exits, we're going to talk about distal colonocytes.
Terrible segue. Jason, you got to work on this. People are going to stop listening to the show if you don't get your game.
All right. This one also in Nature. Distal colonocytes targeted by C. rodentium recruit T-cell help for barrier defense. First and corresponding author, Carleen L. Zindel. Last and corresponding author, Casey T. Weaver. Also published the 10th of April. This one doesn't have the nice little handy thing at the top about saying how long they were in paper purgatory. So I cannot provide that information. All right. So C. rodentium. It is a surrogate organism in mice for E. coli that are they're adherent and effacing. So some E. coli stick to the edge of the cells and then rip them off. That's this type of C. rodentium does that in mice because mice are corpophagic and can eat lots of types of E. coli and not have a problem. But C. rodentium gives them the dysentery. So it targets the middle, the mid gut and distal part of the intestine, what you would think of as the proximal or ascending colon, or transverse, not the distal colon in humans, right? To use that geography. And so what they do is they're like, well, are there differences there? And so they show first and foremost through, guess what, single cell RNA-seq, that in that middle part of the intestine, there's both small bowel-like in the earlier parts of the colon. There's some stuff that looks more like small bowel with some bile acid absorption properties, water absorption properties, and there's also colonocytes that look more distal, even in parts of the colon that are the colon, right? Early in the colon, which we get. We assume it's a spectrum, a gradient, and then it widens up at some point, but not all the transcription factors have gone away. And so, if you encounter some bile acids in the earlier part of the colon, you are going to have some transporters for those. They do a good job mapping that out. And then what they show is that, hey, with a fluorescent E. coli, or not E. coli, Citrobacter rather, you can see that Citrobacter really prefers the distal colonocyte phenotype. So they establish this is what the bacteria infects. Okay. And there's very specific responses to it, and that's where it goes and kills it. And if you knock out the receptor that the bacteria goes on, that goes away. All right. So that's cool. That's not a Nature paper, though, but that's some interesting stuff. There's responses that include colon hyperplasia to push the cells out and keep the bacteria away from the crypts, all this stuff going on. Okay. So after showing and really establishing that these cells are responsible for this, they show that then the colonocytes are not affected by either the microbiome or immune cells in rag knockout mice. Okay. These cells, they find, then respond differently to IL-22, which is a main barrier function, right? Early infections produced by ILCs. Later, though, it's produced by T helper cells. And they're showing this really, IL-22 in this case, really matters in late infection and keeping them alive. So what they then find with the IL-2 knockouts, right, is that they find the kinetic there. What they then find is that the epithelial cells upregulate MHC2. Remember how I said epithelial cells are my favorite immune cell? There we go. MHC2 being presented by epithelial cells, which then signal the T cells later on in infection, 8-10 days in, to pump out a bunch of IL-22, which backfeeds into the system, which causes the cells to both live and undergo the dedifferentiation programs that they see to help get rid of the virus and clear it. And if you kill IL-22 receptor on these cells or you blockade it, you get dead mice. And so this IL-22 is critical to their responses. They demonstrate that Paneth cells and other cells are reverting to stem-like state, which is a thing that other people, including myself, have studied. And so they're seeing that the reverse stemness or reverse back to stem cell state's happening, and that's also driven by IL-22, but only in these distal cells. So that's what makes this really cool, is they link it to MHC, IL-22 through STAT3 activation and crypt protection, but it's IL-22 from T helper cells midway through the infection that really matter. And so that's kind of what got really cool here. They then did some really cool stuff with a novel strain they made with a lymphocytic choriomeningitis virus, LCMV GP66 to 80 epitope. So this is recognized by the SMARTA T cell receptors. So they use these specific, clonally locked in T cell receptors and do adoptive transfer. And then they show through that, it's the T cell recognizing the epitope of the dang Citrobacter presented by the MHC of the epithelial cell that's doing all this. So that's what makes this a Nature paper, right? They knock out the MHC. They use these restricted custom designed bacteria with the custom antigens that you can trace, and they trace the whole thing down to being, MHC with the bug on the T cell if the thing leads to the IL-22, and they show that it's the helper cells, not the ILCs that pump it out early on, that are really critical for this. ILCs early on matter for not just dying right there and then from the innate response, but for that, that second push to really clear the infection with the adaptive system. You got to have the T helper cells pumping out the IL-22 for your gut to fend off the infection.
I love that. I love that that they're presenting a peptide on MHC2. I think, I really like that about these epithelial cells.
MHC2 normally found only on professional antigen-presenting cells, which include enterocytes. Because enterocytes are just really cool macrophages.
Does this apply for all of the HLA, MHC class 2 molecules, like HLA, like DR, DQ, or is there a subset that is expressed?
Well, HLA is the human version, right? So this is the mouse. I think they just
Yeah, you're right. Sorry, dude. Yeah. What
they
is?
just killed MHC2.
Okay. Okay. Yeah. Sorry, I'm thinking of. I always get all the the
I know
nomenclatures.
it's acronym soup, but yeah. But the point being, micey MHC2. I am validated.
Well, but how is it in humans? The same, same,
Well,
same.
yeah, IBD has all these correlations with HLA responses, and it's not just because of it being on the immune cells; it's in the enterocytes.
Yeah. Yeah, that's true. I heard
I
about
mean,
that.
think about it. Gut cells eat your nutrition. Macrophages eat the bad things. Eating is eating in the end.
Okay. Okay.
Gobble,
That's
gobble, gobble. And if you
that's
gobble it
okay.
up, you can see something that shouldn't be there and you can raise a flag. So why would you get rid of that?
Yeah,
Especially
that's
if you're
true.
like living on an environment covered in bacteria.
That makes sense. All right, so it's a nice. Well, so what I see then is a nice teamwork. The helper cells, the enterocytes, they're all just um, working together to keep the barriers in good state.
It takes a team to keep that barrier in state. I agree.
All right. So for the last paper of today, I have I think it's a very interesting story about glucocorticoids. So for sure, we just mentioned them today. What a coincidence. Glucocorticoids, anti-inflammatory, awesome. Uh, and this paper is kind of expanding on of our understanding of how glucocorticoids work. I think that was very cool. And maybe correct me if I'm wrong, but I think this is pretty unorthodox. I think this is quite an interesting change. So I don't
Yeah,
know.
so to provide a little clinical context, glucocorticoids, there's always a contradiction. We know they're transcription factor alterators, which I'm not
Mm-hmm.
writing any of the story here, right? So which is slow. That takes like 12 to 24 hours to work. And the old
Right.
adage is that glucocorticoids take 24 to 48 hours 24 hours to work. You're not going to have an effect the day you have them. But that's not what people experience, and they insist that they get a quick effect from glucocorticoids. You inject it in your
Mm-hmm.
knee. It's not just the lidocaine that makes it feel better. They work quickly. You take your prednisone. 30 minutes later, you're wired and ready to go, and you
Yeah.
feel an effect within 4 hours. Or pick something, and
Yeah.
doctors being us arrogant types were like, no, it takes 12 hours. That's just the placebo effect, 24. But a lot of people always describe that it acts quicker than that transcriptional factor, 12 to 24 hours to make the RNA and to make the DNA and RNA and to make the RNA and the protein.
Mm-hmm.
So there clearly was a disconnect between the wisdom and the reality.
Yeah.
And then, Brenda.
Well, let me tell you then, how can this be? What other mysterious pathways are glucocorticoids exploiting to make us feel better? Well, what else but the metabolism of our immune cells? So let's go. Metabolic rewire promotes anti-inflammatory effects of glucocorticoids is the name of the paper published in Nature on the 10th of April. First author Jean-Philippe Auger, I guess, from I hope he's French and not German, from the lab of Gerhard Krönke at the University Medical Center of Erlangen. And basically, what they see, and I think this is, I mean, knowing a little bit of the fields, like this makes some sense. They find that gluco treatment with glucocorticoids of macrophages um, actually has a kind of a substantial effect on their metabolic reprogramming, and it generate or in results treatment with let's say GCs results in a increase and sustained production of the metabolite known as itaconate, which already for many years we know that is a very well described anti-inflammatory metabolite, and this part of uh, can really uh, regulate inflammation, particularly coming from macrophages. And so this kind of already on itself makes kind of, if you tell me like this, it's like, okay, they produce itaconate. Okay, that I've heard of this itaconate, anti-inflammatory, well known. what they see is that so upon activation, macrophages, one of also the things that we've learned, I think mostly in recent years, is that there is a substantial reprogramming of the metabolism. Activated macrophages and kind of inflammatory macrophages are well known for having a very glycolytic uh, um, metabolism, and they have kind of upon activation, they start changing the way they transfer kind of the carbons from glycolysis to the TCA cycle. They kind of block the TCA cycle to some extent. They shuttle and a lot of these carbons from glucose either to lactate or to fatty acid synthesis or to other kinds of biosynthetic pathways, and this results on the mitochondrial metabolism to be kind of underrepresented on inflammatory activated macrophages. So what they see is that in their experiments, they basically activate macrophages with LPS, and they look what happens when they add GCs on these cultures. And one of the things that they see is that they see quite a considerable change in the uh, uh, mitochondrial metabolism of these macrophages. So they see that they also do RNA. They start with RNA sequencing analysis, and they see a reduction. What is interesting, I think, is that they see a reduction of some specific pro-inflammatory. They do see some changes in transcriptome, but what I think they are mostly interested is in the changes in the metabolism. So they see that whereas LPS can induce a block kind of mitochondrial respiration, this is not. This is to some extent prevented by treatment with glucocorticoids. They see an increase in the number of functional mitochondria, which is lost upon activation usually, and they see that basically the large change when they do carbon. They have this carbon tracing experiments, and they see that the glucose metabolism is kind of redirected, and it's not redirected like it's usually in pro-inflammatory macrophages. Um, and what they see is an influx of carbons into the TCA cycle. And in macrophages, one of the things that is well known is that in very active TCA cycles, one of the as part of the intermediates of the TCA cycle, uh, there is the production of this metabolite called itaconate. So itaconate is a derivative. This is a derivative from succinate citrate. I forgot exactly where, but from the TCA cycle. And there's this enzyme called the aconitate decarboxylase, so ACOD1, and this is the one that produces itaconate. So what they show is that a lot of uh, the anti-inflammatory phenotype and that they obtain from treating activating macrophages with glucocorticoids is lost if you knock out this ACOD1. Although it was important is like what they see is that glucocorticoid treatment did not increase directly the production of itaconate, but it does amplify the production of a generation of itaconate upon LPS treatment. So you have an activation of the macrophage metabolism, but then instead of doing it redirecting towards a more glycolytic metabolite by treating with glucocorticoids, you force or you bring the carbons through the TCA cycle, and then you increase the availability of raw material for generating itaconate. And that's what macrophages do. And this itaconate generates and has a very strong anti-inflammatory effect. So they do a bunch of in vivo models. So they do a LPS induced lung injury model in which they basically put LPS in the intranasally, and this normally results in severe lung inflammation. And GC treatment prevented this. If you treat with glucocorticoids, you can prevent this damage. But this is abrogated if you have ACOD1 knockout mice. So then there's no protection. And this also applies to a model of rheumatoid arthritis in which you inject autoantibodies in the joints. And then again, GC regimen can help, but not if you have ACOD1 deficiency. And they have also a model of allergic airway inflammation with ova in mice. And again, ACOD1 was crucial for the effect of these glucocorticoids in preventing the worst of the disease. I think it's very interesting. It kind of taps into pathways that we already knew. So I think it kind of adds, it seems like a very logical story. But yeah, it definitely has improved our understanding of how this such a highly widely used therapeutic, does its job.
It's super awesome. I saw that come across the news feed, so I'm glad we covered it. And
Yeah,
explains that people aren't crazy.
UMDs, you need to listen to your patients.
Yeah, I know. All right. Well, in addition to listening, we're going to be speaking to Dr. Caleb Lareau at Memorial Sloan Kettering Cancer Center here in just a moment. But before we get to that, this week we'd like to remind our listeners about Cancer Stem Cell News, one of STEMCELL's free weekly scientific newsletters. Cancer Stem Cell News summarizes all the latest research, news, jobs, and events in cancer stem cell research and delivers it right to your inbox every Wednesday. Save time and keep current with Cancer Stem Cell News. Subscribe for free at cancerstemcellnews.com.
For the second part of the show, for our interview, we are joined by Dr. Caleb Lareau. He is assistant professor at the Memorial Sloan Kettering Cancer Center in New York City. And his lab specializes in single-cell multi-omic approaches. And I'm very excited because for me, all of that bioinformatic tools are close to magic. So I'm very excited to discuss about how we can use the power of data and the power of this very high throughput analysis to understand biology and immunology better. So, Dr. Lareau, thank you so much for joining us today.
Thanks for having me on. Big fan of the podcast. It's really an honor to be here talking with you all.
Thank you for being here. All right, Brenda, I started the last one. You can start this one because there's lots of CAR T stuff here.
Well, thank you. That's very kind of you, Jason. I appreciate it. As Jason mentioned, one of the stories we would like to talk to you about is a story that you guys published last year in November, in which you identified the reactivation of herpesvirus in CAR T cells, which I remember at the time was quite I don't know, felt a little bit of an alarm, or I think it was a very interesting story. But I know that you maybe have a way of telling how you reached to this story, but I would like to start with something about using bioinformatics and applying this kind of tools in the right way, and how, especially for those like me who are the children of the wet lab, sometimes we struggle to understand the best ways and to apply this kind of analysis to our research. It feels daunting a bit. So maybe from your perspective, how has your knowledge of bioinformatics really guided your research? Or I know that you collaborate with a lot of people. You can see from your publication list. So that's something kind of fun, something kind of nice that you can do with this knowledge. So what is your experience in bioinformatics and immunology that you would like to share, especially for those who don't have it?
Yeah, thanks for the question. And I think you started on a great topic. I think the reliance on data and the reliance on informatics is what makes our research program distinct from what a lot of other immunologists typically work on. The first thing that I would say is I think that there will be more researchers like me that come out of this generation. I think traditionally it's been difficult to be cross-trained in both cutting edge immunology or molecular biology as well as bioinformatics, just because the fields are so new, especially bioinformatics. I think traditionally you have sort of the archetype of a dry lab collaborator and a wet lab experimentalist sort of putting their heads together to try to figure things out, but really we try to do both, both within our group as well as individuals. Think about how data can actually motivate hypotheses that then can be tested experimentally. And that's really the standard of all of the different projects that we have going on in the group. Start with a large amount of data to really motivate hypotheses that oftentimes are very straightforward experimentally, but you're just asking the question in the right context or in the right light. I think this way of thinking has sort of been traditionally housed under like systems biology in some sense, but now we're really kind of taking it really focused on the immune system. The thing that I always tell people when they ask me, something like what you described as your background, I'm experienced in molecular biology, I'm experienced in immunology, but I want to start getting better at bioinformatics. How do I do that? To me, the answer is not to take a class, and it's not to go back and get a master's degree or anything that. I think the major thing is to identify a problem that you really want to solve. And in doing so, I think you just naturally acquire the skill set to be able to answer that question. And just by setting a very specific goal and kind of running toward it, there's tons of tutorials, there's tons of vignettes, all kinds of information online that allows you to do this. And I think the people that are the best equipped to really pick up this as like a cross-trained discipline in their own research are those that have the strongest conviction about a specific biological question. And so, yeah, I'm very excited to talk about some of the viral reactivation work in cell therapies. But I think that's a case in point. Like, if there's a virus that you're interested in, there's a ton of resources now where you can go online and you can figure out every sequencing experiment that's ever been done on the planet. You can go see where that virus has shown up. And these are now pretty accessible databases where someone has taken the time to make it accessible to anyone with the sort of skill set. But if you have that as a question, then I think you start to unpack it. You start to really understand what goes on in it, and before you know it, you're doing bioinformatics. It happens very quickly.
So, kind of going down this, I know, you know, some people view computers as magic. Some people understand a chunk of it. The more data you start doing, the more magical it gets for some people. Why do you need fancy data pipelines to do this? Why can't I just shove some stuff in Excel and look at things? I know there's a volume problem, right? And I think we all know if you get like a couple thousand rows in Excel, it starts hating you. So there's a little of that. But like, what does it mean to be bringing data science expertise? I think people know what it means when like you go to bioinformatics, they tell you, look, you need 20 mice to get statistical significance based on, they know that power calculation stuff, but this is different. And so what is this new understanding and skill set that's letting us do stuff from the computational side?
I think of it in two different buckets. So the first is just purely an engineering problem. So if I have 50 iPhones worth of data sitting on a hard drive somewhere, how do I efficiently make sense of it all? There's very basic sort of rules in terms of the way that software runs on hardware that then allows you to parallelize tasks, do things sequentially, to take a very simple function and apply it to many, many applications over time. And so this is something you typically learn in a programming class, or you maybe start to get exposure to. But it's really just like the skill set, just like you learn how to pipette or do PCR. You're going to learn just how to talk to a computer and be able to get sort of software executed off the ground. So that's like kind of the first basic skill set. And again, I think you just pick that up from finding a problem that you want to solve. So for example, you want to figure out if a specific 7-mer of nucleic acid is present at what places in the genome. That's like a very well-defined task that you can sort of learn the mechanics of how to interface with a computer, do these kinds of analyses. And the second, and I think this is typically where I find most people to struggle, at least those that have been sort of conventionally trained as immunologists or biologists, is the statistical critical thinking that has to be applied. So it's not just enough to have a sort of the mechanics of being able to talk to data on a computer, but when you go from a structured hypothesis that kind of as you described, you have your 20 mice and you have condition A and condition B, the hypothesis test, the statistical inference is all very well described from the onset. But once you start working with these high dimensional data sets, you're literally generating new hypotheses on the fly. And so it's very easy to fall into these buckets where you may overanalyze something in a way that you think is meaningful, but it actually, just because you're doing so many tests, you actually just discovered noise. So I think it's like those two specific elements. And again, conventionally in undergraduate and some graduate programs, you'll have a programming class and a statistics class where you start to build these intuitions. I think again that if you apply them or you start like analyzing data for a question you care about, you start to get the nuances of these things just kind of as you go. But ultimately, I think things that really help are again having a good problem, but then also having access to individuals that have done it before, and just having some sort of kind of ongoing conversations with them is sort of the things that I've seen be effective. But really, I think it's those two kind of core skill sets. The same way that I could think about uh, pipetting and cell culture maybe being the two sort of key skill sets you need to do to get a basic in vitro experiment off the ground, or how I think about sort of like the core tenets of what informatics means.
I really like that the point that you make that having a problem that you want to solve really uh, motivates you and helps you focus on finding the solutions toward it. So I can totally agree that I think from my experience that's so important because otherwise it's so overwhelming the amount of resources that there are, the amount of different things that you can be doing that unless you're very focused, you get very easily kind of uh, this anxiety that's induced by all the different resources and all the different options. It's hard to choose sometimes, and if you're not focused, then you're just paralyzed.
For me, I think once I got my first taste of real scientific research, it was just impossible for me to sit still in classes, right? Because the things that would be presented to me in lectures maybe weren't as relevant or I couldn't see their utility. And so just being able to like really learn through field work has always been the solution to me. So I think it's both as you're describing, like the focus and sort of the ability to drive progress, sort of with with some focus, as well as just like I think when you are the author of your own curriculum, it just helps immensely go from 0 to 1 to 2 to 3 in terms of your skill sets and these things, and that's just how I've learned myself and how I see strategies be most effective for the trainees I've been fortunate to work with.
So, my last question before we dive into some work that's kind of a test case of this is how much math or computer knowledge do you think is really required for some of this? Because I know people have varieties of feelings about, say, college math courses, and then going and doing analysis they may get nervous about.
Yeah, so I think it's the critical skill is a little less. Did you take differential equations in college and know how to derive one if you know you had to? It's more the sanity checks that are built in every step of the way. There's a ton of statistical theory that has been developed for all kinds of different situations. There are entire fields dedicated to assuming your data looks a certain way, and you know, there's sort of the saying like all models are wrong, but some are useful. That I think is applicable here, where almost there's never a perfect fit for the specific data question you have. Like there's not a hidden equation on page 702 of a textbook that's going to tell you how to like appropriately analyze your data. So the key thing that you want to be able to do is ask yourself, is this signal uh, more than chance? And this chance thing, I think, is the hard thing that sometimes people again feel like there's a right way to define what chance is. And the way that we always do it, or the way that I try to think about this, is what does a shuffle of your data look like? What does a permutation of your data look like? So if, for example, I find 100 viral reads in a particular sequencing experiment, if I were to take a random sequencing experiment, do I also see 100 reads? If I shuffle the people in my data set such that I'm looking at a specific association and then the association is still there, it's this degree of uh, essentially randomness that you may also be picking up on your analyses. Just being able to kind of on the fly think through what is my independent variable, what is my dependent variable, and then can I again permute that independent variable to get a sanity check of what a completely random signal would look like in the analysis I'm doing. That more times than not is the most effective thing you can do, and you don't have to know higher order math. You don't have to have graduate training in statistics to be able to do this. You just have to really use the skills that you already have in terms of thinking about dependent independent variables, and then have the ability to do random permutations, which is one line of code in basically every software environment. If you look at a lot of papers that I publish, I'm doing that far more often than I'm ever doing any sort of like statistical theory that would exceed what is being taught to an undergraduate. And so I think it's like kind of building that skill set and that intuition, which you already have if you're a practicing scientist that can, I think, really unlock some of these informatic analyses.
I love that point that you're making, and it's kind of thinks be smart, like think. My dad would always, as a child, it's like just think, just take a moment to contemplate all the options. And I think I don't know. I guess also in the case of biology, sometimes for us, for when you in a regular kind of standard biological assay, yeah, there's because we don't analyze so much. I think there's a way in which you can kind of with your mind understand what's going on. Like the results usually can kind of grasp what's going on just by just quickly thinking about it. But I think that when you start working with such huge data sets on such small signals or such convoluted type of comparisons, there's just is a different level of thought that you need to put on it and really be a lot more logical and a lot more formal in your train of thought.
Yeah, so as we dive into this virus example, I can give you a couple of examples of
Yeah.
where the thinking about it carefully kind of played in and how we were able to make some conclusions that way.
Well, why don't we jump into that as an example? I think that would be very nice to hear the story from you.
So you have a paper just out in bioarchive showing herpes virus reactivation in CAR T cell therapies, which kind of made a big splash when it hit. So how is big data useful in finding that? Why couldn't you just, I don't know, take everyone who had a CAR T therapy, look at how many people had herpes virus, and then go, oh, this is a problem. Thank you on your clinical readout. I'm done now.
Well, I think the field actually did that second option, and somehow the pieces didn't quite come together. So, a lot of the inspiration for this project originally was reading through the footnotes of as many clinical trials as we could. So, I guess taking a step back, this project really came to be when I was a postdoc at Stanford in Ansu Satpathy's lab, and I didn't really have strong conviction on what my postdoc project was going to be per se. But I generally was excited about how transformative CAR T cells have been, and also sort of recognized that maybe the safety and efficacy of these therapies could still be improved. And that was something that I just really wanted to spend my time thinking about. And so, whenever we found this herpes virus kind of association, the way that we really initially uncovered it was reading through clinical trial footnotes. And to me, that is the single best source of raw material for hypothesis generating in immunological research. Certainly, you can have curious observations in vitro or in murine models, but these are real therapies that have been in real patients, and we're seeing real things that we can't explain. To me, there is no better media to ultimately incite new hypotheses. And so, if you go look at basically any clinical trial that has had CAR T cell therapies administered that say have at least 15 people enrolled, chances are there will be at least one person that will have had HHV6 encephalitis. And so, what this means is that there is a phenotypic presentation where the patient has some sort of inflammation of the brain. It's, as you know, very challenging to disambiguate that which is viral derived versus things like cytokine release syndrome, which is also the sort of more common neurotoxicity in CAR T cell therapies. So ultimately, the way that you have to discriminate these is oftentimes is a differential diagnosis of ICANS, where you will do a PCR for this HHV6 virus, and that's how this HHV6 encephalitis diagnosis will sort of come to be. And so your original question was, why didn't you just sort of look at all the clinical stuff? And the short answer is that we did. The thing that required data is to answer sort of the more basic research question, which is, can we figure out where this virus is coming from, at least in part? And that's where we really needed genomics. Was we already knew that there were patients that were getting HHV6 encephalitis. We just didn't really understand anything about the mechanism or how this could be potentially problematic in a cell therapy setting. And that was really our goal point, was seeing evidence of this already occurring in patients, but not really having an explanation, and then ultimately trying to formulate that into a data science hypothesis that we could analyze.
So just to kind of wrap up this part of the story, what did you find? So once you did this analysis, what were the like thinking about CAR T cells as a therapy. Is there something we can do to prevent this activations? Is there a specific mechanism? It's just why are these viruses being reactivated?
Yeah. So let me span that question with kind of the original insight from the data. So we have this observation from the clinical trial footnotes. Okay, people are getting HHV6 encephalitis from uh, receiving CAR T cell therapies. So it's like, why is this happening? Like, where is the virus coming from? Was kind of our original question. So there are tools online now, and there's one in particular I want to give a shout out to. It's called Ceratus. So this was published by that's also a very cool story. It's actually a team of independent researchers during COVID wanted to ask and answer the question: what are all viruses that have ever been sequenced before by humanity? And so they literally took every public sequencing data set on the planet and they created this giant matrix of every sequencing experiment by every virus just to get the background, just to say, okay, these are things that we know, these are viruses we know, and these are where they are at. Once you get rid of all of those, then they found other nucleic acids that look like viral reads, and they were actually able to describe hundreds of thousands of novel viral contigs purely from data science analyses. This is a Nature paper from, I think, 2021. Again, the resource is called Ceratus. My point being is that this was really the heroic data science piece, and now there is a web portal where you don't have to have any sort of specific informatics training, that you can go and query whatever virus you're interested in and ask the question, where in the history of human sequencing data does this virus show up? So for us, that was really the kind of key first step, is that we have this association with HHV6. Where does this show up? So you do a very basic, you can spend 2-3 minutes on your couch searching HHV6 in this database. And the thing that you'll find is that the single highest level of this virus in a human cell setting where there was no expectation that the virus was there is in primary CD4 memory T cell cultures. So what this means is that there was some experiment that was done. It was actually several experiments over different continents, over different years, where they just went and cultured memory CD4 T cells and did basic things like single cell, or sorry, just bulk RNA-seq at the time. And unbeknownst to the authors, there were sky-high levels of HHV6 in these just standard T cell cultures. So this is all something that, unbeknownst to the authors, just from sort of a systematic reanalysis of the data, you can find largely just from surveying this in a couple minutes of querying this database. So this was actually kind of the first major piece of evidence that we had for our story, is that independent of CARs, independent of any kind of engineering, if you just go and culture memory CD4 T cells, more, not every time, but I don't know, somewhere on 5-10%, depends on how you do cultures, but there will just be the reactivation of this virus. And so your question was, what's causing this, and how do we think about stopping it? What we think is happening is that downstream of TCR signaling, either directly or just through proliferation cues, the virus will start to reactivate as a function of, again, either TCR signaling or proliferation. The way we think about it is, these viruses are maintaining lifelong latency, and they're really just waiting for the right moment that they can poke their head up and see if they can establish a productive reinfection. When they choose to do this is still something that mechanistic virologists are studying, but for us, whatever kind of artificial setting we're putting these T cells in vitro is at least sending some of those signals that these viruses are saying, hey, you know what, I'm going to see if I can become a competent infection again. And in some settings, that indeed happens, and you see lots of this virus reactivate, lyse the cells that it reactivated in, and then ultimately infect other cells in the culture. And so all of that was just sitting there, sequencing data from, say, 2015, 2016, that just culturing cells in vitro, T cells in vitro, specifically memory CD4 cells, could lead to high levels of HHV6 reactivation.
So you saw the observation clinically from the notes, but you couldn't quite get a signal from it. do you think the power is really combining everything, and so you could make a pseudo trial with the higher end that you can never do on its own? Or do you think the power also comes from the fact that you can deep dive the raw data and just like see that it's there? And the hard part is that without this omics stuff, it's needle in a haystack scenario. And so you can't get there.
You need both, right? Like the clinical observations are the clinical observations, whether the genomics data was going to be helpful or not. Like that's still real things that are happening to real patients. But by analogy, there's all kinds of viruses and all kinds of weird cell cultures. It's not all signal, right? There's definitely going to be people coughing on their cell culture, and that leads to signals that you can't explain, and it may not be as relevant. But both having the genomics deep dive that clarified that this is very high signal, and so just again for perspective, like at the end of a 2-week culture, whenever the original authors did RNA-seq, it was something like 4 to 5% of all RNA molecules in that culture were actually viral derived as opposed to human derived, right? So these cell cultures were actually just HHV6 factories by this 2-week period. So that context you need the genomics data for, but that by itself doesn't really mean a whole lot, right? Like having someone stumble upon that as a result without knowing that, oh yeah, like people that receive uh, adoptive cell therapies that also are engineered in vitro go on to develop sometimes lethal complications due to this virus. Like you needed that piece of evidence too to really start to understand why this is a meaningful finding. And so I think yeah, again, you need both. Like there's so much data and there's infinite questions you can ask. How do you know you're asking the relevant ones? I think you orient, or at least the way we do it, is you orient yourselves to things that are actually happening in patients in the clinic, and then ultimately to ask mechanistic hypotheses. That's where we turn to the DNA sequencing and the genomic sequencing data for that.
I have a follow-up question that I guess is a little more mundane, but thinking about so this herpes virus, latent herpes virus, was it from a previous infection to the same individual, or do we have like latent herpes virus already in our genomes and we just pass it on?
Yeah, it's an awesome question, and it's one of my favorite factoids of HHV6. So, for clarity, I did not plan to do my postdoc in HHV6, but there is this one really, really cool fact about this virus. So, in humans, this is the only virus that we know of that can be transmitted through the germline. Meaning, if you are infected, there is a non zero chance that your offspring will then have this virus in every single cell of their bodies. So the mechanism here is the virus infects cells, it can integrate into the telomeres. And if the infection occurs in your germ cells, then those will get passed on under Mendelian rules to your offspring. So there's about 1% of the population is sort of our best guess globally that actually has this, where they have what's called inherited chromosomally integrated HHV6. So these people have at least one copy of the virus in every single cell in their body, irrespective of T cells, irrespective of anything else. So that 1%, you absolutely have the virus in your system. For most of the donors that we were working with, and for most of what we think we've been reanalyzing, we think it's the second case, which is 99% of the population, where you get infected by this virus, usually in the first 1 or 2 years of life. You develop a pretty mild rash, but then your immune system just controls the virus and it clears. Um, the virus, though, then can lay latent in cells that express CD134 or OX40. So this is the canonical HHV6B receptor. HHV6B is the strain that's endemic in North America, Europe, and Asia. Uh, that molecule is most highly expressed on activated CD4 cells. So sort of your rough mechanism is you have T cell activation as a function of an infection. You have these herpes virus in your system. Virus will bind to CD134, ultimately infect via that. Then those activated cells become memory cells when you have this now herpes virus in them that is potentially integrated into the telomeres to establish latency. So there's both mechanisms by which the virus can appear in your T cells, both either acquired through the germline, which again is about 1% of the cases, Or for the rest of us, and again, it's an endemic virus, like basically everyone's been infected. So it's literally like the other 99% will have it in some subset of T cells, just depending on how that infection and latency establishment worked.
Uh, I have so many thoughts. First of all, I'm horrified by the fact that 1% of us have that, and that, and also like then every time you get your, then if you have this infection, you have this latent infection, then every time you have an immune response, are you potentially just getting making more herpes virus? So it's I'm sorry, I'm just like thinking like the implications of having this. It feels a little bit
Well, so
terrifying.
so so here's the good news is that this is not a major global health concern right now. There
Yeah.
are reasons that people are not devoting their entire lab to HHV6. By and large, we really do control the infection. We control latency. We can control reactivation. Our immune system is incredibly proficient at dealing with this ancient pathogen. So, uh, don't your take home from the conversation so far should not be, oh, I found a new virus that I should be super scared about. It's all good for the most part, just in very specific settings. And it's particularly in individuals that are receiving lymphodepletion, say if they're receiving a marrow transplant or a CAR T cell infusion. This is where it's potentially problematic and can be much more, lead to much more complications that can be severe. So it's fascinating biology, but just as much as the virus has found all these kind of clever ways of propagating, our immune system has also found a way to control it. And for most of us, this won't be a problem.
So HHV6, you gave the example, not a global problem, but like something that affects people in specific scenarios. The one I think about that a lot of us get that then comes back later is herpes zoster. So do you think there is, and we kind of know people get chicken pox, now we have vaccines to stop it. Do you know, especially in the era of vaccination for, herpes, for basically chicken pox, as people will think of it, if that's having an effect on the latency rate? Have you guys looked at any of that translatability? Because I'm wondering, like, my generation, everyone has chicken pox and we all have to get shingles vaccines and not get shingles later. I'm wondering in the future if all these vaccines that you got as a kid prevent you from ever getting it, or we're all just still latent and, pop goes the weasel, just like HHV6 type of thing. And that seems like something this type of approach could help answer, because it's a similar question. I didn't know if that's something people have looked into or any of that.
Uh, I would say this way of thinking, and the way that I would describe this way of thinking is we're using kind of unstructured genomic hypotheses to study effectively viral nucleic acids. So the examples that I brought up so far, people just did DNA, RNA sequencing experiments. They see viral nucleic acid there. Or actually, in the original study, they didn't see it there. And then post hoc, people like me see it. That then sort of incites hypotheses. I think this is an extremely new way of thinking. I don't think that there's a ton of other examples of this kind of study. So your point about this, herpes zoster virus, I think these are very relevant questions that we are now probably very like equipped to be able to answer. And this is like a big part of what my lab is doing now. Is we had a good understanding of this HHV6 in this cell therapy context, but other viruses and other contexts in the immune system is kind of a even bigger question, is what we're going after. So I don't have any direct evidence, direct pointers for your specific question now, but I think this way of thinking, like, hey, this is generationally different and this could be having some impact on health, is exactly the sort of buy-in question you need to start to go looking at the data to be able to analyze this. And so I think the first thing you would do is just start looking at public sequencing data and see where you can find these viral nucleotides and start to maybe build an association map of where those reads show up relative to other samples that have been profiled.
Well, I think we are reaching the end of our conversation here. It's very, I could ask you so much more because also you have a lot of collaborations and uh, but I think we need to uh, don't kind of finish up a little here and give our listeners a chance to get to know you a little bit better, something beyond your research. So at the end of the show, we like to ask our guest a question. So I hope you're ready for it. One of the questions, I think one of the favorites are, if you were not a scientist, what would you be? Do you have an answer to that question?
It's a great question. I think when I was around 18 years old, that's when I first learned sort of what a PhD scientist was and got my first taste of it. So, uh, I've been fortunate that I kind of set my direction way back then, and I've mostly been able to follow through it. Uh, there's one thing that one profession that I would really, really enjoy doing, and this is, yeah, very, very much not scientific, but uh, every, I think, Wednesday at Stanford this is where I did my postdoc, is when they cut the grass, and I always remember how good I feel walking outdoors, smelling freshly cut grass. And this is something that, like, growing up as a kid, this is sort of my job, on summer months in high school and that kind of thing, was like mowing lawns. The smell of, like, freshly cut grass, just, I think it, like, incites something psychosomatic in me that just is so much fun. So it's really, really hard to say this in New York City because I do not have the same lush green grass everywhere like we did in Stanford's campus. But taking care of lawns, being outdoors, it's so much fun, and I definitely try to do that whenever I can. Whenever I go back to Oklahoma and visit my parents, that's something I look forward to. So that's my answer.
I'm very into lawn mowing personally. I have refused to give up the chore.
Yeah. Yeah, I guess you don't get a lot of that in New York, do you? That's kind of sad.
Yeah, we'll need to move out of Manhattan one day to get that back. But for now,
Oh.
the concrete jungle is a lot of fun too.
Yeah, well, Long Island is not too far. I know there's some grass to cut over there. I think I understand what you mean with the smell of fresh grass. It's like the smell of the rain, when it starts raining and you feel that earthy. So I totally understand. And I also grew up having to cut the grass. So I'm not gonna lie, I didn't enjoy it too much at the moment, but looking back, I should have appreciated it more.
It may all be nostalgia, but it's fun.
Very likely. So thank you. It was such a great conversation. Thank you so much for joining us. Caleb Lareau from Sloan Memorial Sloan Kettering Cancer Center, talking to us today. It was really a great conversation.
Thank you for having me, and very much enjoyed being a part of it.
Thank you for being on.
This brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.